I don't know. Well, it's like this. To start with, I've been assisting Maria's lab at NILU, the Health Effects Laboratory, after I finished my coursework of Erasmus Mundus, European Master of Quality in Analytical Laboratories. I joined the lab to do my thesis, master's thesis, and the work I've been assigned is to assist them in their process of accreditation by the Norwegian accreditation for GLP. At the outset, having been trained in quality management, the analytical methods and data analysis, and in what way I could apply the theoretical principles for this particular field study. That is broadly my work which I have been doing in this particular laboratory. And when I say implementation of quality management principles to in vitro toxicology studies with special emphasis on nanotoxicology, the first step I had taken was a thorough review of the documents necessary for this particular GLP exercise. And I found that there has been a sort of a lacuna with respect to the forms of quality assurance, which is very much a necessity for this particular GLP accreditation. And I had devoted at least some 2 or 3 months of my initial work for the preparation of various documents, especially the forms relating to quality assurance. And as quality professionals, we say that any activity which is not documented is not done. So that sums up everything. That sums up the whole of the quality work. And there's one more thing which I really found very important was that, in fact, there are two aspects. One is the reproducibility and the other is the repeatability. This can happen only when you have proper documentation. In other words, a particular GLP trial might be performed. Like, for example, we saw this particular study of this silica particles. Fine. And then we got some results, we got some report, everything is perfect. Now, the real crux or the real problem arises if there is another party which wants to perform the same kind of study, and then are they able to get the same kind of results? So the key to this particular repetition or reproducibility is the documentation. And I tell you one thing, from the management perspective, the outlook is different. From the scientific perspective, the outlook is totally different. And as a quality assurance, it's very difficult to bridge the gap between conventional science and management practice. As a quality assurance, it's a blend of both the managerial aspects as well as the basic scientific aspects relating to that particular study. And to further my particular approach here, the quality management tools which I had chosen for this particular study was the internal audits. So I had prepared various checklists of relating to this particular studies, and then I tried to exercise them. I made those documents and I distributed those documents to the study personnel, the study associates, and whatever the data I could get, and then based on that I could furnish some reports. And this particular audits which I had performed, I had in fact divided them into 3 phases: the pre-analytical stage, the analytical stage, and the post-analytical stage. And basing on this, I had prepared various quality assurance documents, and I had got the data, and that's how I could give a particular report. This in short is my experience here in this particular laboratory. And apart from this particular documentation work, one major requirement of a quality assurance personnel or professional is that he or she has to develop very good interpersonal skills, because you're dealing with different kinds of people. You'll be dealing with right from the top level of the management to the grassroots level of the personnel working there. You will have to interact with each and every one. You should always keep them in good humor so that you get the required data which you want. And then I tell you, human resource management is really a tough job. That's my conclusion. Thank you very much.
Probably it would be. Do you have any questions for him?
I would just like to endorse what you said, actually, that lots of people have said to me over the years, what do you look for in an auditor? What are the sort of training criteria that because you can't come out of university with a degree in quality assurance because no one's offering it. And one of the best auditors I ever had was not scientific at all, but he had terrific interpersonal skills. I mean, he was really good with people. He actually had been a salesman before he joined my department, and he is still one of the best quality assurance auditors I've ever trained, actually. I mean, because he had that rare ability to put people at their ease. None of us like being watched when we're doing something. It's an examination, and we don't like people. It inhibits the mere fact of being there inhibits people to do their job properly. So one of the great skills is to actually put them at their ease. And I would, over the years, I've come to realise that that's the big art. You can teach most of the rest, but dealing with people is something that is. I had an auditor who was the reverse, actually, who was academically brilliant but couldn't cope with people. She used to run away. If she was ever sent into one of our animal units where the head of department was a bit hostile, and he used to argue with her, she used to run away. So she never got anything done, you know. So yeah, I would agree with that wholeheartedly.
And probably, thank you, probably it would be maybe interesting for you to know which kinds of handling practice we have in our lab, or we set up in our lab. At least at the moment we have to have some procedures which we follow, and Anya will tell you something about that.
So, welcome everyone. I see that someone still survived. So, I will say a little bit about GLP, but from different side. From side person who need to work in the GLP condition environment, and how it look like when you need to work according a lot of documentation and stuff. But first, I want to present, welcome at you. My institute is in Norway, in here close to the Oslo. If you have some topic, problem about atmosphere, pollutant in atmosphere, climate changes, they are the best people, and they can answer for every question. And in this institute 5 years ago, development health effect laboratory, when I have fantastic occasion to work now. So a few words about my place to work. Our lab is very small, only 4 rooms. Our topic, nanosafety, nanotoxicology, genotoxicology. We analyze also uptake, characterization, repair study. And now you can apply also to come to our lab and have some training, some research. And let's talk about GLP. What we are thinking about GLP, what our quality direction and people who establish for our methods think about GLP. They think they will help us. They make our life easier. But what people who work normal today, every day in lab think about GLP? What really it means GLP for us who need to work? People who create the system not work so much in lab. It's a little look like they try to create the system behind our back, where they based on the information from the article or from some handbook, and it's a little bit complicated. We try to establish method which will improve quality in our lab, improve safety, because it was a big problem in our lab, but also will be useful and friendly, user-friendly, which will be easy. If we create very complicated system, people will not follow them. So try to make some experiment with nanoparticles in our lab. If I order some nanoparticle to test it, I need to make 3 things before I start work. I need to register. So I need to fill some templates, some folder, put a lot of information inside. Only few I put here, but we need to put size, how the site was measured, which method, from which place I have this nanoparticle when they come to our lab, what danger, how handling procedure should be established for this. I need to put some information also in the container, chemical composition, size, also origins. When I put this all information, the small flask, I need to close in special places. Only people who work in the lab have access to these places. So when I make these 3 things, I can thinking about to make experiments. Before I start experiments, I need to ask on this 4 question. If I know where I can work with the nanoparticles, how I can protect myself and people who work at the same time in the lab, what is the danger connected with the nanoparticles, and what is the most important, where I can find information. You can imagine that someone new come to the lab, they have training, everything is work. Suddenly they work alone and something broken, something starts beeping, you cannot find something. You need to have very easy access to every information. You need to know where is information, what you need. So we need to personal training. If I have answer, if I know how to answer for this 4 questions, I can start work. The big problem was about safety, how we should safety handling with the nanoparticles in the lab. So we are looking for information in the publication. A very good paper we found. They said that we have 3 safety level for the nanoparticles. Every time when you want to handle with nanoparticles, you need to find your level. A little complicated for us, so we convert these big graphs to our big graphs. We decided we have only 2 levels safety, one for nanoparticles in suspension and one for nanoparticles in powder, or for the situation where we prepare a solution by using sonicator. So we decided that we have 2 kinds of nanoparticles handling. When we handling with the nanoparticles in suspension, we need only protect skin. We decide to use only nitrile gloves, 2 pairs, because there are some studies that talk that small nanoparticles can introduce that 1 pair is not enough, so we decided 2. But more complicated was the situation when we want to handle nanoparticles in powder, or when we want to prepare solutions by using sonicator. We need to protect our respiratory system, so we need to use mask, HEPA mask, goggle, lab coat, nitrile glove. But what happens if people want to work in the same time in the lab? You have very small space. So we decide that we don't want to look like this man, because it's very complicated and not fashion. And we decide to work in the glove box, which have HEPA filter, and inside there are sterile condition also, so if you prepare some suspension, will be sterile. So we decide to work in closed environment. The last problem what we have is, we start talking about this, we are environmental institute. We know that nanoparticles are danger, so we cannot throw away waste contaminated by nanoparticles. You'll be surprised how many people doing like this. I asked a lot of people how they handle nanoparticles in the lab. All suspension going to sink. They know they are danger, but they don't collect them. So we decided all suspension going to one container which is marked, and when we fill the special special company, we will take care about this. And we decide that every tips, pipette, Falcon tube, every step, a single use, will be contaminated by nanoparticles, will be also going to the risk waste. We decide now to put the line for which waste would be go and for the risk and what for the normal waste. We're still talking, now we put 100 micrograms per milliliter, but we'll see in the future.
But actually, Anya, go back. This is your older slide because everything what is higher than 100 micrograms of risk and lower, less than 100 micrograms per mil, we consider as a normal case.
Sorry.
So it's very high level of protection because 100 micrograms per mil is very low dose. But still we offer this line.
Is that for all material, all parts of these?
Yeah, we decided not to make it. Anya can answer.
What? Sorry, Matthew, can you?
Is this figure of 100 micrograms per mil, is that for all composition of different particles?
Yes, because when you test something new, you got some new nanoparticles, you don't know still about effect, so you don't need to collect somewhere. Check the results, okay, they are not danger, I throw it for the normal waste. We decided everything going contaminated by 100 micrograms per milliliter to risk waste. Yes?
I assume you don't have carbon nanotubes, low carbon nanotubes in your lab.
We will.
We will have.
We will.
You will. Then I would consider that you check if you want to stick with these numbers, because they would be too high if you deal with long carbon nanotubes.
Yes.
Because I think the disposal method probably takes into account there, because with the carbon nanotubes you can have them incinerated, so it's probably better to, irrespective of the volume, just incinerate everything that you have with it.
Yes, in our labs, we work on the carbon nanotubes and incinerate everything. There is a bag where we put everything which is contaminated by carbon nanotubes, because this concentration is very huge. We don't work at this concentration. It works at.
Yeah, maybe in our lab we never work with such toxic substances like carbon.
No, we have a titanium dioxide and PLGA.
Or silver, they are not so.
Yeah, but that's the provisional, just to implement some practice. To start this, there is this paper of Crosso which has these 3 levels, and it's quite useful, but at the moment we have so many new implementation, new practice, that to keep a kind of easy way to distinguish between risk and not risk. But actually we don't have this, we use much less concentration than this in our experiment. This is stock solution, usually.
Yes.
Yeah, it's just an explanation. The Grosso paper, we developed this method for all of EPFL, which is like one of the large engineering and chemistry schools, one of the important schools in Switzerland. So it's for all of the schools, and it's obvious that if you're like a sub-department, then basically you can reduce it to the part that concerns you. Just the person setting up the scheme has to keep in mind that if something will continue, you might have to add branches.
Yeah, I agree. But at the moment, in our level of, we have about 6 different types of nanomaterials, and none of them we found very toxic, definitely not genotoxic. So, nanosilver micro.
Can I ask you, because I mentioned earlier on that this is a general obsession with the inspectors about what you do with your waste. I mean, I don't know why they're so obsessed with it, but probably because they've found so many problems in the past, I would guess is the reason. What do you do with the HEPA filters in the glove boxes?
Oh.
It's it's just new thing which we implemented now, so we need to make some procedure.
Is it dealt with by you, or is it
No, we
dealt
always
with by an
ask-
external company
Yes,
who comes
we have
in
an
and checks
external.
the functionality of it and they change the
Yeah.
filters? So it becomes their responsibility what to do with it, because this is an issue, of course, because if you're using the glove box as a means of protecting the operator, which is essentially what it's for, the regularity with which you change the filters needs to be, to some extent, validated, and you need to have a safe means of disposing of filters which will be contaminated by a wide variety of contaminants.
Yeah.
And it's often cheaper, easier, less hassle to let someone else, it's another SCP, it's someone else's problem. It's often easier to get a contractor to come in and deal with that. And lots of people do.
We actually have a company checking also our HEPA filter in laminar box, and also all waste, risk waste is dealt with the company. We explain what that is risk waste, and they deal with that.
This is a very good point. If you are purchasing new equipment, make sure that your filters can be changed without you having to clean afterwards the entire lab. I visited one big lab, which is otherwise top, and they have a single closing film. That means basically you open the filter box, you take out the filter, and then only you can enclose it. That means your entire lab is contaminated, basically. You're contaminating all of your lab. Basically, they have to empty the lab totally, have the crew come in and change the filter, clean the entire lab, and then repopulate the lab.
Right.
So be careful with the equipment and test first if you can do the change without. There are these systems that have multiple slides that isolate the filter from the system that you can remove, and then you open again the slides. There are these systems on the market. It might be cheaper to buy a new system than to operate the existing one that brings you all this hassle.
I mean, the more expensive the equipment these days, the more likely it is that you will have some sort of maintenance contract offered to you. And that's true for laminar flow cabinets, as it is for analytical equipment, as it is for pneumology analyzers. My advice, generally speaking, from an economic perspective, is let someone else take the strain, rather than risk comment, criticism, or contamination. Take the advantage of, if it's offered, a maintenance contract, which means someone else will come and deal with it and check things and leave you a certificate to say they've done it, which is all that the regulatory authorities are required to see in the end. They don't necessarily expect you to have to do it all. On the subject of waste management generally, it's as well to think about other sorts of waste as well, because as I said this morning, it's become very big business in Europe. We had a problem in France about 10 years ago because the only organisation that can deal with old batteries is based, I believe, in Switzerland, and they went on strike about 10 years ago for better money, and the whole of Europe was paralysed because waste batteries were building up to humongous levels because these guys, I think they got their demands fulfilled very quickly as a consequence. But when I looked into the whole business of how we dealt with waste at my CRO, I think we had to use 12 different waste disposal companies, all of whom are specialists these days. If it's got blood in it, it's potentially it's called hospital waste, so you've got to get somebody special to get rid of that. Organic materials, gases, tissues, cells in culture, any waste from the autopsy area, especially in our case when we were dealing with primates. The risk of contamination from autopsy in primates if you don't dispose of the bodies. Somewhere on the site where I used to work, there's a mass grave which occurred before I ever got there. They used to bury the monkey bodies on site, but someone conveniently lost the map of where they were buried. So one day somebody will dig a large hole in the ground to put another building up and discover to their horror there's several hundred skeletons in there. You wouldn't be able to do that nowadays. So waste management, look very carefully about how you're going to deal with it and how much of an involvement each individual staff member has, and that the way you deal with it is consistent across different departments. I went to a firm once that had, I thought I'd do this as an exercise, was to look at their SOPs on waste management just to see if they all kind of hung together, and the problem was that they didn't. Different departments had different attitudes. One department, for instance, said, if it's a dead body, I need it to be in a clear plastic bag so I can see what it is. And another department said, if it's a dead body, I want it to be in a black bin bag so that nobody can see what it is. And you had the guy who went round picking up all the different bin bags and the waste. He was apparently supposed to be in two places at the same time in the day. It was a minefield, total disaster. So look carefully at this, because this is something that the regulatory authorities, having got bored with looking at everything else, have become totally obsessed with in the last 5 years, and they spend an inordinate amount of time on this.
Yeah, I just only want to add that that's absolutely true what you are saying. In our institute, NILU, we have a very good chemistry lab, and they are certified for ISO. So they have waste management, they have sort of the practice how they deal with waste, and we just join their practice.
Yes, good idea.
It's absolutely for us easy. And also it's one company and there is no problem, so we collect together the waste, and that's only.
There is one more point when you have these safety guidelines, that you can use your brain, it's allowed, and you can deviate. Because think of your health and safety, it's about your health and safety. So there are not just the nanomaterials. So in certain cases you might say, okay, I handle liquids that contain nanoparticles. I'm not supposed to have ever contact, but there are a couple of operations that are quite critical and to do it really right. So you might opt in this situation for taking single surgical gloves, cost a bit more, but it's single glove. They give you a very fine touch, and you can do the operation the way you need. You are protected against accidental spills in those gloves. The chance that something gets through is quite small, but you gain the tactile skill that you might need for that sensitive task, and you protect yourself from doing stupid things and making an accident.
This is what we worried actually. The more protection, more prone to new accident. Sometimes, yeah.
I'm just wondering about that. Is someone responsible for that safety question, like in the lab or not? Because I know that example because I worked in that lab, and I know that was because the PI decided that he wanted to protect the people. But there's some other labs where professors just don't care.
I think it's a difficult issue, because the trouble is it's all very well for management to say, well, we'd like you to do that, but it's a recommendation. There isn't, in many cases, in certain countries, any legislative weight which allows, I mean, when I worked in the UK, we recommended that people who worked in the pharmacy area wore safety glasses. We couldn't force them to. All we could say was, if you get something in your eye, don't come to us for compensation because we gave you the opportunity to put the right stuff on. And in most cases, you can only recommend that people adopt it. It's up to their conscience as to whether or not they feel that is an imposition or not. Most of the time, people should realise that you're trying to do the best for them. Most of the time, if it comes from the top end, if management says, you will do this or you will get fired, then people tend to do it. You know.
But to be honest, it doesn't matter whether your institute is running under GLP or not. In each institute who works with some chemical issue, there is a responsible person for the safety of the workers, and there is a responsible person for the waste management. And so each worker is obliged, it's your responsibility as a scientist to inform yourself, regardless whether GLP or not, what you do. So you have the, actually you are obliged to ask the safety person for your personal protection, you go there. You have to know the name. Actually, if you don't know even the name of those persons, your PI has done something completely wrong. Because at least you should know the name of these two persons, and you have the right and also the obligation to ask them what to do now. And they will tell you what they think is the right way in your institute. Or you discuss together, maybe you can make suggestions. As a scientist, you are educated, you know better what you are working with. But in each institute, there is somebody.
Maybe Elise can give something about the legal situation.
I was just going to say that even though I wasn't here for the whole discussion, the words I don't hear are the legal ones. And even in a case where you have no statute and you have uncertain guidance, it is very important to think about two legal concepts. One is called due diligence, and the other is, as you mentioned a million times today, the chain of custody. In the event that something catastrophic happens at the end of these tests or at the end of the first uses of how these tests are applied, the next thing that will happen is people will search around the world for the place to put the blame and charge the money to fix what's wrong. And the two things is if you're in that chain of custody, you're as likely to get hit with the request for information from a court or an enforcement authority, whether or not there was an underlying law, or to actually be held liable. And the only way you can get out from under is to demonstrate what in the law is called due diligence. This is a concept that exists in many legal systems, even though it's drafted in different ways from one place to the next. And due diligence says, I knew there was a problem because I did all the literature search. And the literature search showed that these were the key problems. And these were the areas where science didn't know what to do. So in an abundance of caution and prudence, in order to achieve the goals of my work, I was obligated to take on a precaution, which I knew there was a problem, but I also knew there was no clear right answer. And therefore, I specifically took the following precautionary steps. I made sure everybody was trained. I made sure all the cabinets were locked. I made sure we wrote to the companies and asked for their certifications to be forwarded to us in writing, which we kept in a file. And we used those certifications to self-validate when we used the incubator to make sure the temperature was correct. We sent the thermometer to the company.
You have set your criteria for
Voilà.
whatever is perceived by a risk assessment process to be areas that you think are not covered legislatively, so you'll make your own rules. But you must make the rules up that are sensible, scientifically, and ensure that they're being applied consistently.
And that they're defensible.
And that they're defensible.
That, to always have the idea in your head, how would I explain this to a third party, correct me if I'm wrong, who's not a scientist, a smart person, a sophisticated person who is ignorant of what we're doing here in this place, but to whom this must make sense, so that even a bad outcome can be explained. Because we did what we could. Who knew? Yeah. We did the search, we asked the experts, we got the certifications, we documented our steps. Here, prosecutor, you can open the file and see every step we took. Please kindly tell us if there was something more we could have done. What more could we have done? And that will save you.
It will, because you've done, as you say, due diligence, you've done as much as you could reasonably do, aside from saying, well, this is obviously dangerous stuff, we don't know how to deal with it, so we won't deal with it, we won't advance science, you know, we won't bother to deal with it. You have to have a strategy.
The one point that you have to be extremely careful. Showing that it's unknown. You should not be afraid to show that there's uncertainty and it's unknown. But that is not permission. That is not a license to then go roughshod and say, well, we'll try anything. We'll cut the heads off the animals 3 days early and see if they bleed.
We'll tip it down the sink because it won't matter.
What'd you say? Tip it down the sink. Exactly.
Sink.
It's not permission to do just anything. But if used appropriately, it does justify the acts you took. And if the acts look sensible in their context, given the background of uncertainty, not the abstract, oh, there's never going to be no risk. The very precise, this was the problem, this is what we were asked to look at, this is how we tried to address it, these were the possible outcomes, these were the steps we took to prevent these other horrible scenarios, then you can defend yourself. And it takes lots of paper.
One further thing I would say is beware that the legislation is evolving as well.
Absolutely.
When I first moved to my facility I worked in France, we had an incinerator which dealt with about 50% of what we produced. But because Clean Air Acts, Europe-wide and specifically in France, evolved, that incinerator became essentially redundant, because in order to make it comply with the legislation as it had evolved, we would have to have incorporated a brand new incinerator with a much taller chimney and a two-phase burner, and that was effectively far more money than we were prepared to spend. So we outsourced the waste disposal thereafter. I mean, the legislation doesn't stay still. It will catch up with you.
Yes, and the other thing is cost will not be your defense most of the time.
No, I didn't do it because it was way too expensive. It's not an answer. You know.
So don't be afraid to ask that difficult question and document a clear response. If somebody says to you, we always do it this way, don't worry, or we're not going to answer that, don't ask that question, you better document that they said that to you. Am I right?
Yeah. It's called covering your own arse. Yeah.
Well, you can talk like that, but I'm not allowed.
Again, it's SEP. It's someone else's problem.
Don't worry in the specific situation to bring up literature like the papers you just showed, or websites. Go to your manager, go to the safety manager. If your institution has one, they should have one. They must have one usually. In most countries it's mandated. And ask them, we are dealing here with these substances. I can't make a professional call. These people recommend that we deal with this. What do you say? And document this approach. Make it by email.
And whatever ultimately is decided, you need to write it down, because if management decides to say, we're not going to do it because it's going to cost too much, then it's their call, not your call. They must be full, and as soon as you say, right, I'd like you to put that in writing and sign it, so I put it on file if anybody ever asks me, all of a sudden they get very nervous and, start, maybe, and you think, yeah, because they don't like, they're very good at pronouncements, but they're not very good at documenting those pronouncements. So, if you look at GLP, it will tell you what management's responsibilities are. In amongst it, it says providing the necessary resources for people to do their job properly. To me, part of that is giving people an adequate working environment.
The only other thing about this is you should be very careful. It feels counterintuitive. In the tough moments when it matters, when you ask the question and they say, well, why are you asking that? Or you shouldn't be asking that. That's the time when you need to document. And a very prudent thing would be to write a memo to file to oneself saying, on this day, I asked for this training program, I asked for this documentation, it was not provided. Because those are the very things that at the time, the short range cost looks like you could lose your job, you could be disciplined, people won't like you because you asked hard questions. The long range cost is if you're caught in this quagmire, you can't get out easily, and it can ruin your career if it goes the wrong way. So do not be afraid to document what has gone on in those moments when it sounds like it's the moment you want the least to do with it and you want to run away.
Because it will come back and bite you. I guarantee it.
Obviously, I'm thanking you because I want to.
Actually, that was a long day, and I think that that was a long and productive day, and I really am very happy that you were so responsive and interactive and discussed so much these issues of GLP that I probably would like, we have still to talk, but I don't think they will add anything new to this topic. You will get them on your CD, so, and if you have any question or any interest of what we were addressing this Isolde, she was basically, highlighted some key questions on quality and also on nanoparticles toxicity and basic problems. And I also wanted to tell you about our experience, but all we were discussing during these days. And only thing I wanted to add was in my talk that actually we can take this spirit of GLP to our research. You don't need necessarily to have a GLP lab, but you could have good practice, good laboratory practice, make your protocol clear, easy to use, make your templates, validate your assays. I wanted to tell you about validation exercise we performed with COMET assay, and as Yugander said, there are pre-analytical steps, analytical steps, and then inter-laboratory validation, and also the other step of validation is in vitro methods, EQUAM validation, and then OECD recommendation. But every lab, even research, should adopt the methodology or develop methodology, standardize, validate it in own lab, and it's really your task to do and perform the best practice in your laboratory from start to the end, from planning to the performing and to recording and monitoring also what you did, because that will save your time. You don't need to repeat that many experiments. You will know what is going on, and you can also identify what is maybe sometimes basic results, because sometimes we have so many variation and we don't understand why. So, would you like to add something?
I just wanted to point out that GLP is kind of growing fast, and the OECD family is getting bigger all the time. I mean, India were admitted to the OECD early last year. Malaysia is probably the next country that will be incorporated. One of the big areas of research that doesn't get touched by GLP, or didn't get touched by GLP, is medical research in third world countries. And about 10 years ago, the World Health Organization decided to look at how you can make people do good basic medical research in third world countries without losing the overtone of what GLP requires. And they came up with a document which was called QPBR, Quality Practices in Basic Biomedical Research. It should actually be called QPBR, but presumably the two B's were complicated. What it essentially means was the World Health Organization got a whole bunch of quality professionals together, including the lead inspector in Switzerland, me, several other people, a guy from the FDA. The woman who ran this program thought we were going to come up with something that she called GLP light, which was kind of like GLP without caffeine in it, I think. And she was vastly surprised when we all said, no, that's not the way we should go. We shouldn't make it easy for people just because they don't have inspectors and because they live in Peru. We should be expecting people to adopt the same quality standards, so that the day will come when they find a miracle cure for malaria based on some rare plant that grows on top of a mountain in the Andes. But the research they've done was done to a basic quality standard that will be acceptable. They won't have to repeat all the work. And this has grown into a document which the World Health Organization has now produced for that kind of thing. But if you read it, you'll find that much of what it contains is GLP. It won't let you let go of the basic principles of GLP, which are, you must be trained, you must write things down, you must standardise your approach, you must have a plan of what you're going to do, and you must own up to mistakes. Those are essentially the five, I'll boil it down so you don't have to go home and read it and become insomniac. The basic key issues in GLP are relatively few, but they are vitally important, and it doesn't matter whether you work in an institution, a university, a contract lab, or whatever. you know, there is a basic underpinning requirement in doing good science for all of those things. I don't think anybody would argue with any of them. The only thing that GLP came up with when it was written in 1974 that people thought was not terribly kosher was quality assurance. It was the only thing in GLP that people had never heard of before. The rest of it was just basically good science. If you're going to do good science, you need to write things down. You need to make sure you do it the same way every time. You need to be good at what you do, trained. Pass the information on to other people via some documented references, such as standard operating procedures. It's not new. It's the way science has been done since the 19th century. If it's been done properly. So none of this is new. So even when you try to apply GLP to third world countries, they're still being asked to do all those basic things. So, you know, just because you're in a new technological area does not remove from you the requirement to do science in what is traditionally the right way. You know? So, the take-home message, I think, from today, if there is one, get on the OECD website and get those monographs downloaded, because they're all good fun to read, and they tell you a hell of a lot more about how you apply it on the ground. Clearly, waste disposal is a big issue, isn't it? And writing a protocol that contains the information that is enough for somebody to do the job, but put all the standard stuff in SOPs. Yeah? That's all I want to say. Yes?
I would like to ask a question. So, for me as an institute or person looking at the institute, what is the difference if we have an accreditation according to ISO? Basically, we have exactly the same stuff. We have SOPs, we have a QA system, we have to document, we have to write a study report, we have to do exactly everything perfectly the same. So where for me is the difference whether I'm accredited according to GLP or according to ISO? Where is the benefit for me to go for GLP if we have an ISO?
The short answer to that is the American response to it is, and to a large extent it's not untrue, GLP is an obligation. You have to be inspected under GLP by your local national inspectors if you declare yourself compliant.
Well, we have to do with the ISO system.
No, you don't.
ISO
you pay for. The difference is that GLP inspections are a requirement. ISO is optional.
It's an option, but we need audits for it.
Yeah, all right, but you can.
And you have to pay
It's
for the
something.
audits.
The Americans. The Americans do not accept ISO as a quality standard for the simple reason that when ISO first hit America, hundreds, literally, of people set themselves up as independent ISO inspectors with no qualifications whatsoever and accredited facilities as being ISO compliant. And when the FDA discovered that these laboratories were generating data in some cases which was finding its way into GLP.
But the situation is different in Europe, so I would like to have an answer.
Well, it is and it isn't.
I just want to say two things, because this really is a subject for a whole separate lecture that it would be
Or another
joyous to
day,
give.
probably.
The first thing is, I agree that the origins of the authority are very different, and perhaps the actual text of individual procedures or protocols, whichever word you prefer, are very different. For the person in the lab who does not make the decision which should govern and etc., and just has to obey a bunch of rules in order to obey their job requirements, that's a very tough problem, and it seems to me in the future there will be moves towards harmonization, and in the short term, it's a problem. Not everything has a straight, clear, pretty answer. And it's a problem. And it's a problem the field needs to know about and think about and worry about and push for harmonization. The second thing I wanted to say, because we're going to run out of time, is thank you. Because Hazel and Michael have been slaving for weeks, if not months, to put together an absolutely splendid week. And I think we ought to thank them. And I think that today's work was incredibly courageous, which usually doesn't get much credit, and very kind. And their effort is to take people and bring them to a new level of thinking and in a new way of thinking. And I think that both of them are outstanding professionals and wonderful role
We have one more question before we get to Pakistan.
Wait, let's just thank you first.
No, please, keep the discussion going. Guys, can you have that conversation so we can all hear it? Kurt, can you tell us what you're saying? We want to hear what the answer is.
Well, the question I had for the lady was, if she talks about ISO, what does she mean? And I have now the answer.
So can we get some resolution on this so that we're not all
that
that
is an important issue because in Europe ISO accreditation is something very common.
No, wait. The ISO is many things, but you have to say which ISO.
Maybe you say it.
I think you mean ISO 17025. So you cannot be accredited to ISO. You have to specify the documents. So we're not talking about ISO 9001, which is a certification and which is way less cumbersome than.
What would the difference be, for instance, if you were to declare to the German authorities? And Germany is a very peculiar place.
Well, we are a German authority.
Sorry?
We are the federal German authority for risk assessment. And that's why we have to do the accreditation. We have to be accredited.
Yeah, but why can't you be GLP instead?
Because GLP is much more narrow than ISO 17025.
No.
So in some extent.
It's simply that GLP applies only to safety
Exactly,
studies.
yeah.
And if you don't work in that field, you can't be GLP because no one will inspect you.
Exactly. So it's very similar.
I mean, to give you another example, clinical laboratories that perform hematology analysis for clinical patients, but it's basically just the hematology.
They have the same accreditation as them.
They have the same problem, which is that the GLP authorities won't have anything to do with them because they're not working on animal samples. But because they're not analysing the blood for drug levels, the GCP authorities won't have anything to do with them either. So their short option is to go ISO. And often that is the answer. If you can't get your local inspectors to include you in the programme because you're not.
In Germany, it's not that you have whatever ISO accreditation institute running around. So accreditation institute for ISO needs to be certified on its own. Yeah? It's not that there are 100 around. There is one which is responsible for us. One.
Sure,
But, yeah,
that's okay.
To make it brief, make it brief. I have listened very carefully. I've looked through all the documents. It's perfectly the same as with ISO.
Oh, absolutely. No, I couldn't agree more.
Can I just check with you, Andrea? Is it the same person, the same body in Germany that accredits ISO?
No,
it's not the same.
No.
The standards are the same. Okay.
But I don't see the benefit as I don't, not that I want to be mean also, I just would like to understand the difference.
There isn't much. I mean, there are a few tiny differences, and one of which is the legal obligation of the study director to sign the final report and stand up and defend that in court if he has to, which you don't with ISO. But in terms of what they require you to do as working practices, there's very little difference between them. As I say, the only difference being that if you perform work which will find its way into a preclinical submission dossier, you have to be GLP compliant. If you don't or can't, then ISO's your best bet. And that's the only, I mean, if I sent work to you as a subcontractor, for instance, I wouldn't try and make you GLP. I would declare you in my study plan as being compliant with ISO. Any quality standard will do for me, because it's better than nothing.
Please, if you say GLP, then ISO is not the equivalent. The equivalent of GLP is ISO 17025. It's not the same.
No, I agree.
So if you talk about OECD, you need to say ISO. And if you say GLP, you have to say 17025.
You have to specify
which ISO you're talking about.
The accreditation you're talking about. Okay, okay. Perfect.
That was very interesting
Then
discussion.
I would like to just say a few thank yous to people, if that's okay.
Only one thing I want to say that in Norway we have Norwegian accreditation agency and it's for both ISO and GLP the same. Of course different department but same organization. And in our institute we have chemistry lab ISO and we are applying for GLP and we adopted many procedures already adopted in ISO lab. But basically we saw difference that GLP is a process because all is inspected, but in ISO it's method.
Yes.
And this is why it's not necessary to sign in ISO all study because you have method.
Perfect. Okay, well then I have a few short thank yous to make. First and foremost is to all of you for participating and for making this a really lively discussion and for making me not have to give another talk. So you saved me that effort. But having said that, I think Maria and myself we might talk to Jacques afterwards to see could we get our presentations, could we record ourselves the presentations so that we can link them up for you so that you can at some stage yourselves hear what we would have said. But we're not going to keep you for another hour just for that. So you have the slides, or you will have the slides currently, and we'll get the voiceover to accompany them offline rather than making you sit through another hour. Then I would like to thank, of course, my co-organisers, Maria and Inge. So Inge is not here, unfortunately, but Maria, come and give me a big hug and a kiss.
Thank you. It was a pleasure.